Hello friends, welcome to Biology Made Easy. Today we will discuss the structure of DNA. The DNA is a macromolecule. It consists of long chains of nucleotides, and the nucleic acid was first discovered by Frederick Miescher, but what is its actual structure, it was given by Watson and Crick. So that model is very famous model, and how they found out that model, now we are going to discuss. According to Watson and Crick, the structure of the DNA is a double helical structure. But how they came to this conclusion? It is based on two experiments. The number one experiment is that of Wilkins and Rosalind Franklin. They suggested that the DNA consists of a single helical structure. But during the same time, another scientist, Erwin Chargaff, he studied the chemical analysis of DNA and came to the conclusion that number one, in a DNA, the amount of purines is equal to amount of pyrimidines. That means A plus G is equal to C plus T. Second one, the amount of adenine is equal to amount of thymine, or we can tell A divided by T is equal to 1. The amount of G is equal to C, or we can tell G by C is equal to 1. The third one, A plus T divided by G plus C is not equal to 1, but it is constant for a particular species. These three are known as Chargaff's rule, which is very important for examination point of view. So when Watson and Crick found that according to Wilkins and Franklin, it is a helical structure, and according to Chargaff, the amount of purines is equal to pyrimidines, the amount of adenine is equal to thymine, amount of guanosine is equal to cytosine, and this is not correct. So it came to their mind, unless a DNA is a double helical structure, this is not possible. Why the amount of adenine will be equal to thymine? Why the amount of purines will be equal to pyrimidines? So they suggested their famous model, that is three-dimensional structure of DNA. And according to them, the DNA is a double helical structure. They modified the single helical structure. But how to prove it? This was given to Wilkins to prove whether it is actually single-stranded or double-stranded. And Wilkins experimentally proved that actually it is a double-stranded structure. So ultimately in 1962, Watson, Crick and Wilkins, these three were awarded Nobel Prize for this model. Now we have to discuss what is the different structure, what is the model, what are the details of this model. So according to Watson and Crick, the DNA consists of a helical structure of two polynucleotides. And in these two polynucleotides, the backbone is made up of sugar and phosphate. Sugar, 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 sugar. Now you see, this is phosphate, this is phosphate, this is phosphate, then this is phosphate, and again they are connected. So this is one. In the other side, this is sugar, 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 sugar. The phosphate is present like this. This is connected like this. So always the phosphate is present in the 5 prime side and sugar is present in 3 prime side. That means this is running from 5 prime to 3 prime. But you see this side, the sugar is present in the 3 prime and here the phosphate is present in the 5 prime. So in one polynucleotide, this is sugar phosphate backbone, and in other there is sugar phosphate. But one is running from 5 prime to 3 prime and the other is running from 3 to 5 prime. And the nitrogen bases, they project inside. This is the double helical structure. And these two polynucleotides, they are always anti-parallel, means they run always in opposite direction. As already have told you, one is running from 5 prime to 3 prime, the other is running from 3 prime to 5 prime. And in the phosphate of one, it's replaced by the sugar on the other side, and vice versa in this case. So again, between these two polynucleotides, the nitrogen bases are paired. How? The purine of one is connected with a pyrimidine of another. Now this is very important. Purines are usually double ring structures and pyrimidines are single ring structures. So suppose here there will be double ring, here there will be a single ring. That means there are 3 rings. Here again, one purine and pyrimidine. In all cases there will be purine and pyrimidine. That is why the white will always remain constant. Otherwise it would have been zigzag. And again, this base pairing, it confers a specificity to the DNA. Because always A will pair with T by means of 2 hydrogen bonds. And what are the 2 hydrogen bonds? Is 6 and 1, and here it will get 4 and 3. That means 6 will combine with 4 and 1 will combine with 3. Suppose here it is guanine and cytosine. They will be connected by 3 hydrogen bonds. And here what are the positions? 6, 1, 2. And here 4, 3, 2. That means 6 will combine with 4, 1 will combine with 3, and 2 will combine with 2. This is very important for all higher types of examinations. So this base pairing confers specificity, and as a result, if the base pairing of one polynucleotide is known to us, the other can be easily predicted. For example, suppose in one 5' and 3', the nucleotides are A, G, C, T, A, like that. Then what is the sequence in the other? You can easily guess. In other case, it will be just anti-parallel. So 3', this will be 5'. Here it is A, here it will be T. G, C, C, G, T, A, T. They are always predicted. From this point of view, Watson and Crick thought that probably there is a semi-conservative replication. Initially they have proposed, though they have not proved it. And these two base pairings, they coil in a right-handed manner always, so that it looks like a coiled ladder. If you have marked some iron ladders are present in front of some subs where there is no sufficient space, how it runs? In a limited space it runs like this. And the sugar phosphates, they form the backbones, just like you hold the 2 sides. And the steps are just like the ATGC base pairs. And for a complete pitch, it takes 34 angstrom. For a complete pitch, means for a 360 degree angle, it is 34 angstrom. The pitch is 34 angstrom. And within this pitch, there are 10 base pairs. That means the distance between 2 pairs, consecutive pairs, is 3.4 angstrom. So within 360 degree, there are 10 base pairs, and the gap between them is 3.4 angstrom. Now you think of a coiled ladder, and suppose there is AT. How the next one will be there? The next one is GC. How? 3.4 degree height and 36 angle. Then next one again 3.4 and again 36. So when 36 will come 10 times, it will complete 1 pitch power. And when this 3.4 will come 10 times, it is 34. This is very interesting. Again, the distance between these, or you can tell the width, is 20 angstrom or 2 nm, you can tell. So this is the basic structure of DNA. It is double helical, it is right-handed, they are anti-parallel, they confer specificity. If one is lost or destroyed, the other can be synthesized, it can be predicted, and the pitch is 34 angstrom, in which 10 base pairs are there, and the distance between two conjunctives is 3.4 angstrom, and the width is 20 angstrom. These are the major characters. From this you can get so many questions for examinations, because the amount of A is equal to T, G is equal to C. So suppose in question the amount of cytosine will be given, then you can predict other ones automatically. Suppose the length of the DNA is given and you will be asked how many base pairs are there. Very easily you can do, because the gap between two is 3.4 angstrom. So simply you divide by that and you will get how many base pairs are there. Suppose you will be asked how many coils are there. In one coil there are 10. So again you divide by 10. These are the common questions that are asked. And again between G and C there are 3 bonds. A and T 2 bonds. Suppose you asked in a DNA there are 300, 400 bonds are there, and the cytosine is 30, 40, 50 like that. Then what is the other one? You can calculate from these bonds. Simple mathematics. So this is the general structure of the DNA, which usually we call it as B DNA. But in addition to this, there are also some other forms of DNA like A DNA, C DNA and Z DNA, and this variation depends upon the method of isolation, the relative humidity, the ionic strength. All these things are there, and accordingly this A can be converted to B, B can be converted to D. All these things are there. But the Z DNA is completely different. The Z DNA is usually found in E. coli. That is not right-handed, that is left-handed like this. And that is not helical, that is zigzag. So I will give you what is the difference between these different types of DNA molecules, A, B, C and Z. Just you see it. So now you see here one table is given, A, B, C and Z DNA. And what is the difference between them? The main difference you see lies in the condition of relative humidity. In A DNA it is 75%, B it is 92%, C it is 66%, while Z DNA is found in high salt concentration. Similarly, A, B, C are right-handed, while Z is left-handed. And again you see the base pairs per ton, per 360 degree ton, it is 11, it is 10, it is 9.33, while here 12 base pairs are there. Similarly, the vertical rise 2.5, 3.4, 3.3, but here it is 3.8. And similarly, you see the white diameter, here is 23, here 20, here 19, and here 18.4. And commonly this B DNA is found in our cells, which we have discussed in detail. Now we have discussed the structure of DNA. Now what are the important functions? Number 1, the genetic material, or you can tell all the hereditary material, the codes, what type of proteins, what type of enzymes will be synthesized in cell, all this blueprint lies in the DNA. That is number 1 function. Now the second function, if there will be a slight change in one of the nucleotides of DNA, then it will lead to devastation, means mutation. That is why the pairing between the two polynucleotides is very important. Third function, autocatalytic function. Autocatalytic function. What is the meaning? One DNA has to produce two DNA during the S phase of cell cycle. So that is known as replication, and that is an autocatalytic function of DNA. The fourth is heterocatalytic function. In this case, one DNA will produce one RNA, and that RNA will carry its activities like synthesis of proteins and then all those things. That means the DNA have these four very, very important functions. Now, I will give you few questions. The first question, if in a DNA the amount of thymine is 30%, then what is the percentage of cytosine? Very easily you can answer it. I am giving some choices. A choice 30%, B choice 20%, C choice 40%, and D choice 60%. Then second question. In one DNA strand, the sequence is 5 dash A G C T A G 3 dash 3 prime 5 prime to 5 prime. Then what is the sequence in the other one? A choice. Now 5 prime T C G A T C 3 prime. B choice 3 prime to 5 prime T C G A T G. Next one 5 prime. 5 prime, this will be G A T C G A. D choice is 3 prime to 5 prime. It will be C T A G C T. It will be very easy, you can easily answer. The third question is again very, very important. Which is the correct statement? A choice. A plus T divided by G plus C is not equal to 1. B choice. A plus T divided by G plus C is not equal to 1. Then third choice you can write A plus G divided by C plus T is not equal to 1. D choice, none of the above. Means that nothing is correct here. You will be able to answer all these questions, very easy questions. After going through the DNA structure, it is very easy. The structure of the DNA is very important for all types of examinations. I hope you have understood it perfectly. So thank you very much for watching the video. Again, we will meet in another video. So wish you a very good day.